Hi, hello, good evening everyone. I'm Dr. Harika Kishore. On behalf of Vista's Learning, I'm here as your biology educator. V-Learning, it's an online education platform which works with an ideology of providing quality education for all. Visit our website www.v-learning.in where you're going to get an access for all the live classes, recorded sessions, and also study materials. And moreover, you're going to get this lecture notes in the form of PDFs. Make sure to get your acquaintance with all the channels that are available and subscribe to the channel Vista's Learning Class 11 and 12. If you find this session useful and interesting, make sure to click on the like and share this session with your friends. And also click on the bell icon such that you are going to get all the upcoming video notifications. Saying that, let's start our today's session. Today's session, we are going to start with a new chapter, and that is Molecular Basis of Inheritance. It's a chapter 6 that comes under the unit Genetics and Evolution. We very well know about the importance of this chapter because it is not only important state board wise, and even it is very important NEET wise. There are like 4 to 5 questions every year that do appear from this chapter, that is molecular basis of inheritance. So it's very important to give a special attention towards this chapter, and that is called as molecular basis of inheritance. So here, as we already know that inheritance is nothing but the transmission of the characters or the traits that comes from that passes from one generation, that is parent generation, to the progeny generation. This chapter is all about made you to understand about the study of genes, genetic inheritance, genetic material, and also genetic code, and even we are going to study about the human genome sequencing, right? So before understanding all about the factors which are responsible for storing the information that has to be passed from the one generation to the another generation, it is very important to know about the genetic material. See, we have learned about the inheritance. Inheritance is nothing but heredity, transmission of the characters. How the progeny looks similar to their parents, or the progeny looks different from their parents. That is all about inheritance, if the characters are passing from the one generation to the another generation, and variation, if the characters which are not present in the parents, if it is going to present in the future generation, that is the progeny generation. It's all about the principles of inheritance and variation. In what way the characters or the traits are passing on from the parent generation to the offspring generation, in what way they are not carrying, what way they are variating from their parent generation. It is all about principles of inheritance and variation, right? So heredity, where we find the passing of both physical as well as mental characters or the traits that happens from parent generation to the progeny generation, that passes from one generation to the another generation. So here, inherited patterns and the genetic basis of such patterns. At the time of Mendel, where we come across with the experiments that were conducted by the Mendels on a pea plant. So there, the nature of those factors, they call them as a factors, because at that time, we are not aware of the DNA which is acting as a genetic material in almost all the majority of the organisms. And this DNA is responsible for storing the information which is necessary to pass on from one generation to the another generation. This DNAs are the information blueprints. Then, that means at that time, we are not aware of this genetic material. So that is why we call it as a factors. They are those factors that have the capacity to regulate. Regulate what? Regulating the pattern of inheritance. Regulating the pattern of inheritance. That is a heredity, genetic inheritance. That is a passing of the characters, passing of the traits from one generation to the another generation was not clear. It is not clear at that Mendel's time, but now it took so long time over the next 100 years, around we can say 1856. This is the time 50 to 56, Mendel have conducted his experiments. And after that, almost it took 100 years where we can find Watson and Crick's double helical structure of the DNA was proposed in the year 1953, right? So over next 100 years, the nature of the putative, repetitive, very famous genetic material was investigated, culminating, that means with the highest known developments in the realization that DNA, what do you mean by DNA? DNA is a deoxyribonucleic acid. Deoxyribonucleic acid. We have nucleic acids, we know that we have two types of nucleic acid. One is DNA and other one is RNA. In that, DNA is acting as a genetic material, at least for the majority of the organisms. Majority of the organisms has this DNA as their genetic material, by which it stores this information and by which it passes the characters trace from one generation to the another generation. We have learned that nucleic acids are the polymer of nucleotides. See, while we are discussing about biomolecules in the class 11 biology, we come across so many biomolecules like carbohydrates, proteins, fats, nucleic acids. There in which we come across with terms like monomers, polymers, right? So, look at here. If you consider this as a monomers, monomers combined with each other by means of bonds and leads to the formation of polymers. See, if these monomers, in the case of carbohydrates, they are nothing but monosaccharides. Each and every monosaccharides combined by forming glycosidic bonds in between them and leads to the formation of polysaccharide. Monosaccharides combines to give rise to polysaccharides. That means monomers combines to form polymers. In the case of carbohydrates, that are monosaccharides combines to give rise to polysaccharides. If you come with an example of like protein, in the protein, amino acids will be your monomers. And each and every amino acid combined by means of peptide bonds to form a protein structure, complete protein, and that complete protein is your polymer. Same way, if it comes to the nucleic acid, in nucleic acids, these monomeric units are nothing but nucleotides. In carbohydrates, monosaccharides. In proteins, amino acids. In nucleic acids, they are nucleotides. Each and every nucleotide combined together to form polynucleotide, that is nothing but your DNA or RNA. So that is what nucleic acids are, the polymers of nucleotides. Then each and every nucleotide is nothing but monomers. Each and every monomer combines to give rise to polymer. Each and every nucleotide combines to form polymer of nucleic acids. Now, so here, as we already said, nucleic acids, DNA, RNA. And these DNA and RNA are made up of monomeric units and that are called as your nucleotide. Nucleotide. See, every nucleotide will have three components in it. What are they? Nitrogenous base or nitrogen base, sugar in the form of pentose sugar, that is ribose, and phosphate group. Nucleotide, there is a small tip to remember. See, most of the students will get a confusion between nucleoside and nucleotide. Nucleoside is a combination of nitrogen base with sugar. It have two components in it. Nucleoside combines with phosphate, then it forms nucleotide. So, nitrogen base and sugar, we call it as a nucleoside. S for sugar plus nitrogen base. Okay. So nucleotide 3, tide T for 3. That means it consists of 3 components. One is nitrogenous base, nitrogen base, sugar, and finally that is phosphate group. So we can say that nucleotide is nothing but nucleosides plus phosphate group. So here DNA acts as a genetic material in most of the organisms. We just now mentioned that majority of the organisms will have the DNA as the genetic material. Then what about RNA? RNA also acts as a genetic material in some of the viruses. For example, like retroviruses and even coronaviruses, HIV and even hepatitis. These are all some of the examples of the viruses which have RNA as its genetic material. Now, RNA, though it acts as a genetic material in some viruses, mostly functions as messenger. See, DNA, if you look at the central dogma of molecular basis, DNA will produce its another copy, exact copy by the process called as replication. We know that. Replication is nothing but a biological process by which the original DNA produces its daughter copies. That is, the DNA is producing its copies. So DNA transcribes into mRNA, and this process we call it as a transcription, right? Transcription. Now, whatever the information which is present in the mRNA, actually mRNA is acting as a messenger. Thereby, it is carrying the information that is present in the DNA to produce, to synthesize protein. And for synthesizing this protein, as we already said that proteins are made up of amino acids. The sequencing of the amino acids, which amino acid has to come after what amino acid, this information will be presented by the mRNA, and this mRNA is transcribed from the DNA. That is what we say, the DNA will store the information needed which is useful for passage of the characters from one generation to the another generation. Now, the process by which the protein synthesis takes place by using the information that is present in the mRNA, and that process is called translation, right? See, not only RNA is acting as a genetic material in some of the viruses, but also it is acting as a messenger. That is what we call it as an mRNA. It is acting as a messenger. Even it is, besides being acting as a messenger for the transfer of the information, and thereby it is useful in the synthesis of the protein, also RNA has an additional roles along with being acting as a genetic material and also useful as a messenger. It is going to act as a catalyst, and also structural functions it has, and also adapter. Look at here, it functions as a adapter, it has structural functions, and in some cases it is going to act as a catalytic molecule which is going to accelerate the rate of the reaction. Now, we have learned structure of nucleotide. Structure of nucleotide. Just now I said that. See, nucleotide, remember this 3 indicates that, this T indicates that the nucleotide will have 3 components in it. This is just a mark to remember. So here, what are the 3 components that are present in the nucleotide? One is nitrogen bases, nitrogenous bases, and sugar. Finally, that is phosphate group. Right? So here This sugar is pentose and that is ribose. Here, ribose will be present in RNA and deoxyribose will be present in DNA. Now coming to nitrogen bases, we have purines, and pyrimidines. We have purines and pyrimidines. Adenine, guanine comes under purines. Cytosine, uracil, thymine comes under pyrimidines, right? So we have a pentose sugar, that means 5 carbon containing sugar, and that sugar is ribose. The ribose will be a sugar in RNA, whereas a deoxyribose is a sugar in DNA. That is what we got the name like deoxyribonucleic acid. Now, so here, and the way these monomers units are linked to form nucleic acid polymer. Just now I said that the monomer units link together to form the polymeric units, that is polymeric nucleic acids. Now, so here in this chapter we're going to discuss about structure of DNA, replication. Just now we mentioned one DNA first it get uncoiled off. Now each single stranded DNA, that is parent DNA, will act as a template for forming daughter DNA, right? Says that it finally leads to copies of DNA. So towards the starting, we have the DNA, and that DNA will get uncoiled. Each one strand will act as a template for the formation of the another strand, and finally it leads to the formation of copies of the DNA. That is what we call it as a replication. While we come to the replication, we are going to deal about the conservative method, semi-conservative method, like this. Now, after that, process of making RNA from DNA, process of making RNA from DNA. And that is what we call it as a transcription. So the DNA, whatever the information which is present in the DNA is now going to be transcribed into mRNA. And this process is called as transcription. Right? Now, the process of making RNA from DNA, that is called as transcription. Now, the genetic code that determines the sequence of amino acids. Just now we said that amino acids combined by peptide bonds to produce protein. And the sequencing of each and every amino acid that comes from the genetic code, that will determine genetic code that determines the sequence of amino acid in the protein. These are the sequence of the amino acids which are going to come to produce protein, that is protein synthesis. The process of protein synthesis, just we know said that translation, right? Translation. And for all this process, there is a regulation. There is a checkpoints to check whether the process is going on fine or not, whether there are any errors present or not. So the elementary basis of their regulation. Regulation is always needed for this processes. So here, formation of the DNA, that is replication, and the DNA is transcribed into mRNA. That process is called as transcription, and this mRNA information is useful to synthesize the protein by sequencing the amino acids, and that is called as translation. All these things we are going to deal in detail in this chapter. Along with this, we are also going to study about the human genome sequencing, the determination of complete nucleotide sequence of human genome. That is what human genome sequencing during the last decade has set in a new era of genomics. Study of genome, study of genes of an individual and its interaction of one gene with another. That will all comes in your genomics. Now, in the last section of this chapter, the essential of human genome sequencing, why we need to sequence the human genome, the essentials of human genome sequencing and its consequences will also be discussed. Before that, we need to first focus on the structure of DNA. Let us begin our discussion by first understanding the structure of the most interesting molecule in the living system, that is DNA. As we already know, DNA is going to act as a genetic material in most of the organisms, and how it is going to show its relationship with an other nucleic acid, that is RNA, how the DNA relates itself with an RNA. So here, in subsequent section, we will understand that why it is most abundant. It is means DNA is acting as a genetic material, abundant genetic material, and what its relationship is with RNA. Now, let's understand about the structure of the DNA. The DNA, it's a long polymer of deoxynucleotide. Then the monomer units are, we already know, monomer units are nucleotides. It is a polymer, long polymer, which is made up of deoxyribonucleotides. Why we are calling it as a deoxy? Because the ribose nucleotide has sugar, and that sugar is a pentose, ribose. At the second position, it lacks oxygen. That is what we consider it as a deoxyribose. Deoxyribose. Deoxyribose sugar is present in the nucleotides of DNA. That is what we call it as a deoxyribonucleotides. Now, the length of the DNA, this is very, very important characteristic of in each and every organism. The length of the DNA is usually defined as a number of nucleotides, And here, the pair of nucleotide is referred to as one base pair, which is abbreviated as BP. Pair of nucleotide refers to as the base pairs present in it. The length of the DNA is usually defined as a number of the nucleotides present. As we already know, monomeric units are nucleotides. How many monomeric units combine together to form the polynucleotide? That is a long polymer of deoxyribonucleic acid. That is the length of the DNA. Now, this length of the DNA is a characteristic of an organism, right? Let's see. These are numericals that has to be remembered. For example, if you take this is a bacteriophage, and again lambda is also a bacteriophage. Here, the bacteriophage known as pi into 174, it contains 5,386 nucleotides. Now, coming to the lambda bacteriophage, it has 48,502 base pairs. Base pairs means pair of nucleotide, right? And Escherichia coli has 4.6 into 10 to the power of 6 base pairs. These numericals are important to remember. And if it comes to human, haploid content of human DNA is 3.3 into 10 to the power of 9 base pairs. This is haploid content. If it is diploid, content will be double, 6.6. So the haploid content of the human DNA is 3.3 into 10 to the power of 9 base pairs. All these numericals are important to remember. This will give the length of the DNA of these organisms. Now, let us discuss the structure of such a long polymer. So here, let us recapitulate. Let's summarize whatever the things we have known so far about the DNA. As we already know, DNA made up of monomeric units of nucleotides. It is a polymer. It's made up of nucleotides. Each and every nucleotide will have three components in it. What are they? Nitrogenous bases, which is divided into two: purines, pyrimidids, right? And sugar. Sugar always present in the form of pentose sugar, that is ribose, right? And the final thing is phosphate group. So the chemical structure of a polynucleotide chain. Let's summarize it. As we already know, nucleotide, right? Nucleotide has three components in it. Nitrogenous bases, purines, pyrimidines, adenine, guanine. Here, cytosine, uracil, thymine. Here is a point. There is a difference between DNA and RNA. Cytosine is commonly present in both DNA and RNA, whereas the uracil will be present only in the RNA but not in the DNA. And thymine will be present in the DNA but not in RNA. This is important. So, uracil will be seen in RNA, thymine will be seen in DNA. Now, so here, the nucleotide has three components: nitrogenous bases, pentose sugar, that is ribose. Ribose in case of RNA, as I already said that, the oxygen is missing, will be not present at 2' of hydroxyl group that is present in the sugar. That is what it is called as deoxyribose, because it lacks oxygen. Deoxyribose for DNA. Ribose is a sugar for RNA. Deoxyribose is the sugar for DNA. That's it. Next, the final component, that is phosphate group. So nitrogenous base 1, pentose sugar 2, phosphate group 3. Three components that are present in nucleotide, right? Now, so here, just now we discussed that the nitrogen bases are of two types, purines and pyrimidids, right? So here, there are two types of nitrogenous bases: purines, adenine and guanine. Adenine and guanine, right? Pyrimidines, cytosine, uracil, thymine. Cytosine is common in both DNA and RNA. Cytosine will be present in both DNA and RNA. This is very, very important, right? Now, the thymine is present only in DNA. Thymine will be present in DNA. Uracil is present in RNA. That comes at the place of thymine. Wherever we find the thymine in the DNA, if it is transcribed to be RNA, then the thymine will be replaced by the uracil in RNA. Now, so this is the single strand polynucleotide chain. We can see that if you consider this unit, right, we have three components. One is nitrogenous base, here that is purine adenine, and ribose sugar, and here phosphate, right? As we find the thymine, this can be considered as a DNA. So this is deoxyribose. So the sugar, nitrogenous base, phosphate. These three components constitute nucleotide. Let's start numbering here. 1, sugar, is of pentose. That means 5 carbons. 1, 2, 3, 4, and this is 5th carbon. While we are numbering the carbons that are present in the sugar, we will use prime. 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. Because the carbon atoms are also present in the nitrogen bases like thymine, adenine, guanine, cytosine, uracil. We name them as a carbon 1, carbon 2, 3, 4 like that. To differentiate the numbering of the carbons that are present in nitrogen bases and the carbons that are present in the sugar, for the sugar carbons, we are giving it as a prime. That is 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. Right? So here, this is how one nucleotide connects with an, here again a second nucleotide, here again third nucleotide. See, we have nitrogen base, sugar, phosphate. Nitrogen base, sugar, phosphate. 3 together nucleotide. This is one nucleotide, another nucleotide, another nucleotide, another nucleotide, which combines by the means of phosphodiester bonds to form polymer, that is polynucleotide chain. Now, so here, this is at one side, right? So, the backbone, this is one strand. If you made the same strand vertical, it will look like this, right? This is one strand. And the same strand will run complementary to this to form double-stranded structure, double strand. Here we can see one is running from 5 prime to 3 prime and the other one will run from 3 prime to 5 prime, complementary to each other. That is what we can say. Here the numbering goes like 1, 2, 3, 4, 5. If you come here, here is 1, 2, 3, 4, 5 because it runs complementary to each other. And the bases which are projecting between these two strands are attached by means of hydrogen bonds. And here we do find the presence of 2 hydrogen bonds between adenine and thymine, and 3 hydrogen bonds will be present between guanine and cytosine, and vice versa. Cytosine, guanine, right? This is about pairing. Now, so here, structure of polynucleotide chain. The nitrogenous bases, see, these are the nitrogenous bases. These are nitrogenous bases, nitrogen bases, is linked to, here we can see, while already said, we use prime for the numbering of the carbons that are present in the sugar. 1 prime, 2 prime, 3 prime, 4 prime, 5 prime, right? The nitrogenous base is linked to hydroxyl group here. Hydroxyl group of 1 prime carbon of pentose sugar, deoxyribose, pentose sugar, and the bond linkage between the nitrogen base and sugar is N-glycosidic bond. This is the N-glycosidic bond. See, I'll show here. Here is nitrogen base. This is your sugar. The link is coming from first prime carbon, hydroxyl group, and this link is N-glycosidic linkage to form nucleoside. Nucleoside, that means sugar plus nitrogen base. When this sugar and nitrogen base combines with a phosphate, we call it as a nucleotide. And this nucleosides, see, based upon the nitrogen base to which the pentose sugar is added, is linked by means of N-glycosidic linkage, will give different names for the nucleosides. If the nitrogen base is adenine, then we call it as a adenosine. This is an RNA. Then what about DNA? DNA is deoxy, no? Deoxyadenosine. If the nitrogen base is guanine, then we call it as a guanosine. These are all nucleosides. Guanosine, this is in RNA. Then coming to DNA, that is deoxyguanosine. Simple. If the nitrogen base is cytosine, then the nucleoside is cytidine. And that is in DNA, it is called as deoxycytidine. If it is uracil, see uracil will be present in only DNA, only RNA. So, uridine, and this uracil is present only in the RNA. But in DNA, we find T, that is thymidine. Thymine, so deoxythymidine. Thymine, deoxythymidine. So, uracil, this is the nitrogen base, find in RNA. Thymine, this is the nitrogen base, find in DNA. Right? Now, so here, this is all about the N-glycosidic bond present between sugar and nitrogen base, right? Now, when the phosphate group is linked to the nucleoside, that is what we call it as a nucleotide. Phosphate group is linked to hydroxyl group of 5 prime. See, again we'll start numbering here. First carbon, 1 prime, 2 prime, 3 prime, 4 prime, and here it is 5 prime. Right, this hydroxyl group. See, linked to hydroxyl group of 5 prime. This is 5. 5 prime carbon of the pentose sugar. 5 prime carbon of the nucleoside. Now, this is nucleoside. Nitrogen base plus sugar. Nucleoside through phosphoester bond. So, this is single strand. Double strand will be there, no? That is what we call it as a phosphodiester bonds. A corresponding nucleotide. Nucleoside combines with phosphate group, gives rise to nucleotide. 3 components. The deoxynucleotides depends upon the type of sugar present. We call it as a nucleotide in the case of RNA, and we call it as a deoxynucleotide in the case of the DNA, because the sugar will decide whether it is a ribose or deoxyribose. If it is ribose, nucleotide. If it is deoxyribose, deoxynucleotide. Now, 2 nucleotide. See here, this is one nucleotide and this is another nucleotide. This is third nucleotide in this chain and this is the fourth nucleotide in this chain. So this is 1 and this is 2. One which have phosphate, sugar, nitrogen base. Phosphate, sugar, nitrogen base. Now, 2 nucleotides are linked through, this is important, 3 prime, 5 prime. See, starts numbering here. 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. So here is the 5 prime and this is 3 prime, right? So 3 prime, 5 prime, 3 prime, 5 prime, right? So 3 prime, 5 prime, phospho diester linkage. 3 prime, so here again I'll number here. First prime carbon, second, third, fourth and fifth, right? This is fifth one, this is third prime. 3 prime, it is linking to 5 prime. So 3 prime, 5 prime phosphodiester linkage to form dinucleotide. That means 2 nucleotide. This is 1 and this is 2. Combined by 3 prime, 5 prime phosphodiester bond to form dinucleotide. In the same way, it connects to another nucleotide to form polynucleotide. Simple. More nucleotides can be joined in such a manner to form polynucleotide. This is a polynucleotide where here we find 3 prime, 5 prime phosphodiester linkage. A primer thus formed has one end a free phosphate moiety. At this end, we find free 5' phosphate moiety, and that end we call it as a 5' end of sugar, which is refers to as a 5' end of the polynucleotide. And the other, we have 3 hydroxyl group, 3' hydroxyl group, what we call it as a 3' end of the polynucleotide chain. Same way, similarly, the other end of the polymer, the sugar has free hydroxyl group at 3' carbon, which is referred to as a 3' end of the polynucleotide chain. Here we find 5' end of the polynucleotide chain, and we find here 3' end of the polynucleotide chain. The backbone, this is backbone. See, we have sugar, phosphate, phosphate, sugar, phosphate, again sugar, phosphate, sugar, phosphate. So this is what it is linked, right? From there, we find bases. If you look at here, this is easy to understand. This is the backbone where we have sugar, we have phosphate, we have sugar, we have phosphate, we have sugar, we have phosphate, we have sugar, we have phosphate. If you compare the structure of double stranded to a ladder, the ladder have 2 strands, and the backbone of the each strand will be made by the alternative sugar phosphate, sugar phosphate, sugar phosphate. In between, we do find the projection of the nitrogen bases, and the nitrogen bases will be combined by means of hydrogen bonds. And the base pair, just now I said that between adenine and guanine, it is 2 hydrogen bonds, and between guanine and cytosine, it is 3 hydrogen bonds. So here, the backbone of the polynucleotide chain, the backbone of the polynucleotide chain is formed due to sugar and phosphates, sugar and phosphate, sugar and phosphate. The nitrogenous bases, these are the nitrogenous bases. Nitrogenous bases, right? The nitrogenous bases linked to sugar moiety projects from the backbone. It projects from the backbone, and that nitrogen bases are connected by means of hydrogen bonds, right? So now, here in RNA, what are the differences? Let's see. Every nucleotide residue has an additional hydroxyl group present at 2' carbon position of the ribose. So here, okay, so this is 1, this is 2, this is 3, this is 4, and here 5. To the 2', we have hydroxyl group attached. That is what we call it as a ribose. And in the DNA, we have only H, deoxyribose, but hydroxyl group will be present in the sugar, that is ribose of the RNA. Also, RNA, in the RNA, the uracil is found at the place of thymine. Thymine is present in DNA. Uracil is present in RNA. And thymine is nothing but 5-methyl uracil. 5-methyl uracil, another chemical name for thymine. Thymine, if we fix the methyl group at the fifth position, then that is called as the uracil. So here 5-methyl uracil is nothing but your thymine. Now, so here DNA is a negatively charged molecule. It is an acidic in nature, present in the nucleus. The presence of the DNA in the nucleus was first time identified by Friedrich Mister. Friedrich Mister in the year 1869, and he named this DNA as a nuclein, as it is present inside the nucleus. However, due to technical limitation, see, the length of the DNA is very, very huge. We can't isolate it intact. But we can't isolate it completely intactly, and that is what it is. The structure of the DNA, the study of the structure of the DNA was remained elusive. We don't know. There is no clear idea about the structure of the DNA until and unless it is done by the Watson and Crick in the year 1953. So, however, due to some technical limitations in isolating such a long polymer intact without disturbing intactly, the isolation of the DNA is very, very difficult. Because of that limitation, the elucidation of the structure of DNA remained elusive. It was not cleared for a very long period of time. It was only in 1953 that James Watson and Francis Crick, Watson and Crick model of double helical structure, right? Based on X-ray diffraction, X-ray diffraction data produced by Maurice Wilkin and Rosalind Franklin, popularly called as Wilkin Franklin, proposed a very simple but famous double helix model for the structure of DNA. They have proposed double helical structure of DNA. To propose this, they have taken the data that have given by the Milken and Franklin, as well as they also consider the proportions that were given by the Chargaffs, right? Erwin Chargaff. One of the hallmarks of their proposition was base pairing, right? So here, base pairing between two strands. That is, just now we said that the purines and pyrimidines, how they are going to base pair, show the base pairs, like two hydrogen bonds present in between adenine and thymine, and three hydrogen bonds are present between guanine and cytosine of polynucleotide chain. So, however, this proposition was also based on, just now we mentioned, Erwin Chargaff's rule. This says that purines will be equal to pyrimidines, and here the percentage of A will be equal to the percentage of T, and the percentage of G will be equals to the percentage of C, right? And always purines will be equal to pyrimidines. So this can be explained like percentage of the adenine has to be equal with the percentage of the thymine. See, for example, these are two strands A, C, like it goes like this, right? As it is a complementary base pairing, on the other side, on this strand, we'll get adenine will be paired with thymine, guanine will be paired with cytosine, cytosine will be paired with guanine, cytosine, guanine, adenine, right? Coming to the hydrogen bonds, 2 here, 3, 3, 3 hydrogen bonds, 3 and 2. For example, according to this rule, what we said that percentage of the adenine will be equal to the percentage of the thymine. For example, here, take at the look of adenine, 1 here and 1 here, 2. That means 2 thymines has to be present, 1, 2. It is equal. And the percentage of the guanine should be equal to the percentage of the cytosine. So here, 1, 2, 3, 4. 1, 2, 3, 4. That is why we say the percentage of the purines has to be matched with the percentage of the pyrimidine. That means that the purines occupies 50%, then definitely the pyrimidines are going to occupy the 50% of the composition. So here, for a double-stranded DNA, the ratio between adenine and thymine, guanine and cytosine are constant. It will be equal to 1. Now, here, the base pairing, right, conforms a very unique property to the polynucleotide chain. They are said to be complementary to each other. Complementary to each other. That means this side, if it is A, this side purine, pyrimidine. Purine, pyrimidine, complementary to each other. And therefore, if the sequence of the bases in one strand is known, then the sequence on the other strand can be easily predicted. Yes, we can. See, for example, this is one strand which is having if you know the sequence that is present on one strand, it is very easy, it is very predictable on the other strand because it is complementary to each other. T, A, G, C, T, G. That's it. So if you know the sequence on one strand, it is very easy to predict the sequence on the other strand. Now, so here also, if one strand of DNA, let us call it as a parent DNA, acts as a template for synthesis of new strand. See, this is a parent DNA which separates into two single strands. One second. See, double-stranded DNA. It separates to single-stranded DNA. Right? Say for example. Okay. As the other strand will have the complementary sequence to this strand, then the base pairing will be like T, A, C, G, T. Now, these strands are acting as a template for forming another new strand. This is new strand now. Right? So here we can say that T, A, C, G, T. Similarly, here A, T, G, C, A. And this will be identical with the mother strand because each one of its strand will be acting as a template for the formation of the next sequence. If you know the one sequence of one strand, we can easily predict the sequence that is present on the other strand. So here also, if each strand of the DNA, let's call it as a parent strand, acts as a template. Here it is acting as a template, here it is acting as a template. For sequence of the new strand, the two double-stranded DNA, let it called as daughter DNA, then produce would be identical to the parental DNA because these are parent strands. Let's take the parent strands here and see, right? So what are they? A, T, G, C, A. Similar, no? That is what, because of this, the genetic implication of the structure of the DNA become very, very clear. See, these two strands, that means that this is the parent strand. Parent strands separates and coil, and each strand is acting as a template for the formation of the new strand. Now the daughter strands whichever produced are similar to the strands of the parent strand because it is following base pairing, complementary base pairing, right? Yeah. This is it for the present class. We discussed about the structure of the DNA. Further, we discuss about the helical structure of the DNA in the next class. Until then, keep learning with relearning. Thank you.